Bismillahirrahmanirrahim. In this video, I'll elaborate the cellular proliferation, another step in the healing process. These are the references for this video. So, cellular proliferation is evoked by various cytokines and matrix. Whenever there is tissue injury, there is initial cellular surge that will lead to transient increase in the cellularity. This is responsible for replacement of the damaged cells and also for the immune surveillance. The transient cellularity is the result of proliferation and migration that will lead to the development of granulation tissue, that is highly vascularized transient tissue. And initially there is accumulation of circulating leukocyte. As we have discussed, there is accumulation of circulating leukocyte, resident cells such as endothelial cell, fibroblast, myofibroblast, pericyte, smooth muscle cells, and then there are local as well as marrow driven progenitor cells with leukocyte like traits that come to this area, and some differentiate into transient epithelial, transient endothelial and fibroblastic cells. Then there are some cells, terminal differentiated cells such as cardiac myocyte and neurons. They do not regenerate and contribute to repair. There is obviously healing by fibrosis, there is no repair. And this cellular proliferation and migration is governed by growth factors and chemokines that are also termed as soluble signals, various autocrine and paracrine mechanism, and mixed matrix derived signals. So the signal transduction mechanism in the inflammation or receptor system, they drive cellular behavior in wound healing. Important pathways, signal transduction mechanism that are involved are tyrosine kinase receptor, where the ligands are peptide growth factors such as VEGF, FGF, and EGF, and they stimulate cell proliferation, differentiation, and survival. And there is the mechanism that they trigger the intracellular phosphorylation cascade that leads to the further action. Then there are some G protein coupled receptors. Their ligand are chemokines and lipids and small peptides. They regulate chemotaxis, inflammation, migration, and angiogenesis, and they use second messenger system for as of cyclic AMP, intracellular calcium, etc. Integrin receptors, their ligand are extracellular matrix components such as fibronectin, collagen, and laminin. They mediate the adhesion of the cell, migration of the cell, and also regulate the mechanotransduction. They signal via focal adhesion and cytoskeletal linkage. So although the the receptors, they bind different ligand, all three systems of the receptor intersect at shared intracellular signaling hubs. The G protein coupled receptor for chemokines and other factor, tyrosine kinase receptor for peptide growth factor, and integrin receptors for extracellular matrix, they converge on MAP kinase or serine threonine kinase pathway, and they anchor cells to extracellular matrix, and they transform the mechanical forces into the biochemical signals. And due to this crosstalk and amplification and reinforcement of signals, the cell behave like they proliferate, they migrate. There are cytoskeletal reorganization, and also the changes in the expression of the gene. So this is the summary of the events that we have seen so far. The three signal transduction mechanism or receptor mechanism, the tyrosine kinase receptor, G protein coupled receptor, that ligand for this is chemokine, while peptides are for tyrosine kinase, while integrin are associated with extracellular matrix component. So all these, they, through the activities of kinases, they transduce intracellular signal, and there will be proliferation of the cells, cytoskeletal changes, cellular migration, and altered gene expression. Thank you for watching this video. If you have any query, please write in the comment box.